Most researchers study cancer only after it's diagnosed. I study cancer before we even know it's there. Tumors don't just appear overnight. We now know it can take years or even decades for them to grow to a size at which we can detect them. Understanding this before period is super important if we want to know how to detect cancer early or even prevent it before it even starts. However, studying this before period can be pretty difficult, especially if we're trying to study it in the human body. This is because, for one, there's no tumor that we can detect that we can take out and sequence or profile. And two, we can't take a healthy person and ask them if we can look at all their cells to see if they might become cancer in the future. So, to solve this problem, I'm studying cancer by recreating this cancer development process in a dish. Now, a lot of other people do this too. But most of the time what they do is they take normal cells and they engineer specific mutations that we already know are associated with cancer and see what happens. I don't want to limit myself to just studying mutations that we already know cause cancer. I want to discover new mutations that maybe we can target to prevent or detect cancer early. So what I'm doing is I am starting with normal cells. But instead, what I'm doing is I'm encouraging them to become cancer on their own, instead of telling them exactly how to become cancer based on the knowledge I already have. So cancer is a disease where cells grow in places or under conditions where they're not normally supposed to grow. So what I'm doing is I'm taking these normal cells in a dish and I'm growing them under harsh conditions where most cells would normally die or at least not grow very well. Now, most of these cells don't do well, but there's a small population of cells that figure out by themselves how to survive under these conditions. They'll expand and create this new set of cells after maybe months of evolution in a culture that we can then take and profile. We can sequence them maybe and see what mutations ended up in that population. Those mutations are probably associated with cancer development because they cause these cells to look more like cancer. So by recreating this process that normally happens in the body in a dish, we can maybe discover new therapeutic targets that cause cells to acquire behaviors that are associated with cancer. Now to do this, we need to start with some normal cells from humans. What we've chosen to do is start with normal human stomach organoids. So what we do to generate these is we take some cells from the stomach lining of people who don't have cancer. So these start out all normal, and we grow them in a dish. And when we put them in a dish, they form these little spheres. You can kind of see some of the cross sections in this picture here. There's a great one on the right side where they grow in these little spheres, and the outside of the spheres are essentially like the stomach lining in a person. And the inside of the spheres is like the open part in the middle of your stomach. But these organoids are great for our study because, one, they come from humans and not from some other animal. Two, they have this interesting 3D structure that better recapitulates the actual biology of the stomach rather than the normal 2D flat cultures that we often grow cells in. And three, we can culture these outside of the body for months or even years. So we can study this prolonged period of cancer development and evolution outside of the body. So let me go through an example of the experiments that we're doing with these normal human stomach organoids. So first we start with normal ones, and they're very happy when we put them in our normal culture conditions. You can see how nice and round they are, and there are many of them. They're doing very well, and part of the reason for that is because we are growing them in these super deluxe accommodations, essentially, and we're giving them a lot of these growth factors. Growth factors are proteins that a body normally makes to tell cells that everything's okay, that you should be growing and dividing, you're welcome here. Importantly, the absence of these growth factors is also a signal that the body can send out to tell the cells, you're not welcome here. You shouldn't survive. You shouldn't stop growing. And often, cancer learns to ignore that signal. It'll keep growing even if there's no growth factors around. So what we did is we took out the growth factors from this culture. And as expected, because these are normal cells, most of them die. They're not doing very well in that picture. You can see that the organoids are starting to fragment. There's much fewer of them. But it turns out there's a very small number of them in these cultures that figure out how to survive under these harsh conditions. And if we wait a few months, they'll actually grow out and take over the culture. And they start to look fairly happy again, even though they're in a condition where they shouldn't normally grow. These cells are kind of now like gastric cancer cells. We know that stomach cancer, one of the hallmarks of stomach cancer is that it often can grow independent of these growth factors. And we've actually created a cell line in this dish that can do the same thing. So how did they figure out how to do that? Turns out they actually don't use one of the mutations that we already know about that's common in stomach cancer to grow under these conditions. Instead, what they've done is they've figured out by themselves how to create their own growth factors, something that they wouldn't normally do in the body. So how can we use this information that we found from this particular example experiment to think about preventing or treating cancer? So one thing we could do is perhaps design a drug based on the changes that we see in these new evolved organoids that targets this specific new ability that would prevent these cells from growing under these conditions where they shouldn't and maybe prevent cancer from happening. More broadly, we're interested in creating a bunch of these harsh culture conditions to select for cells with different cancer-associated behaviors. I've given you this example of being able to grow without growth factors, but we're also trying to create cancer cells that can invade other tissues maybe, or can grow in nutrient-deprived or oxygen-deprived conditions. The results of our studies hopefully will give us a set of new mutations or non-genetic changes that cause these cancer-causing behaviors that we can maybe target therapeutically in the future. Using these, the results of our studies, we're hoping to design new ways to stop cancer in its very earliest stages and prevent it from even starting.